I'm about to now. Go, it's recording.
So there are a handful of topics that go into neural tube development, and that's what we're going to talk about here. Or, well, not development, more neural tube formation. The first one is convergent extension, and what that is the way that cells kind of intercalate with each other and form a long and skinny tube as opposed to like a short and squat one. So what happens is these cells kind of mesh together and form like a long like a long neural tube. So the next thing is planar cell polarity, and what that is kind of how cells know the top to the back, and this is the Jesse, this is anterior, right?
Yes.
Okay.
Yeah.
Pickled pet. I'll get to that in a second.
Okay.
So there's a handful. There's a handful of molecules that go into this planar cell polarity, and they define the posterior side of the cell and the anterior side of the cell. And so for the anterior side, the main thing is that flamingo binds to itself, and it's on both sides. And for the anterior side, there's vangled, which binds to frizzled, and inside vangled, or inside the anterior side, vangled binds to pickled. And then frizzled, which again binds to vangled, binds to disheveled, or no, excuse me, Diego and disheveled. So the next thing is apical constriction. And this is really, really, really important for the closure of neural tubes. And how this happens is there's a band of of actin at the top of the cell, and that's what these lines represent. And myosin make these bands contract by like myosin, the motor molecules, they make the actin filaments contract, which essentially turns this kind of what is normally square cell into a kind of like trapezoidal shape, because it like kind of contracts it, makes it, you know, a different shape. So that's, if all of these cells here are doing that, it ends up making it closed. And going back to convergent extension, if these cells don't intercalate properly, then it won't close, because it's going to be too wide for the tube to close.
That's what you said.
Really? And then the final step for closure is this interaction between adhesion molecules. And so there are these classes of adhesion molecules called CAMs, and within that class is another class called CADs, or cadherins, and that's another class of CAMs. And they're really important for these two different tissues. So all this tissue here that becomes the neural tube is coated in NCADs, as I have here in pink. And then this out here are covered in ECADs, and that's like ectodermal tissue. And what happens is, as apical constriction is occurring, these cells that are normally next to the swap binding partners in order to close that gap. So the ECAD, which is normally next to the NCAD, has to trade between them so that ECAD can join with ECAD and NCAD can join with NCAD. And the way that works is they actually prefer being bound to their same class. So that's like they get close enough such that they bind together. And then
That's just blotting.
Well, do you want to cover blotting in a different video, like when we talked about?
I mean, I think we can figure out snowdrop.
Okay. So there's 3 different types of blotting, and they do different things. There's southern blotting, northern blotting, or, well, I'm going to explain a mnemonic for how to remember what the blotting is for. There's southern, northern, and then O and western. And there's nothing O, it's just to remember this mnemonic. And it spells out snow. And then we're going to spell out drop, and that's going to tell us what each blotting does. So there's DNA, RNA, and then O, which is nothing. And it's just to remember for the mnemonic. And then P, which is for protein. So southern blotting detects DNA. Northern blotting detects RNA. O is just a placeholder. And then western blotting is for protein.